Today we're going to be talking about bacterial conjugation. And bacterial conjugation is simply a process by which one bacteria gives another bacteria a piece of genetic information. And again, this piece of genetic information is going to allow the recipient bacteria to be able to do something that it couldn't do before. Maybe that means make a protein that would protect it against a certain antibiotic. Maybe it is make a protein or enzyme that allows it to use a nutritional source that it couldn't have used before. Maybe it's the ability to make a particular toxin. In any case, what's going to happen here is this recipient bacteria is going to be able to do something it couldn't do before. And that is going to give it a competitive advantage over its fellow bacteria and also over other bacterial species that it would be competing against. So let's go ahead and talk about bacterial conjugation. So what I'm going to do is I'm going to draw two bacteria side by side. So there's one bacteria, and here's another bacteria. We're going to draw the chromosome. Here's the chromosome of one bacteria, again double-stranded DNA, and here's the chromosome of the other bacteria, again double-stranded DNA. And this bacteria is going to have the F factor. The F factor is essentially a plasmid that contains some particular piece of information. So let's go ahead and label that. That's going to be the F factor. And again, it's essentially a plasmid that contains a piece of information. Because this bacterial cell has an F plasmid, we're going to refer to it as F positive. And because this one doesn't, we're going to call that F minus. Now, these two bacteria can locate each other through chemical mediators, and they know that one has it and one doesn't. So one bacteria is going to share this F plasmid with another bacteria. And usually these bacteria are of the same species or they're closely related. And again, by sharing this piece of information, this bacteria is ensuring that its own species or a closely related species has a competitive advantage over the others. Now, let's go ahead and draw another diagram and continue our story. So I'm going to redraw these two bacteria. Let's go ahead and draw the chromosomes here. And this F factor contains information to make a pilus. And a pilus is a tube that's going to connect the two bacteria. A pilus is used for attachment. Bacteria have pili, or a pilus, which is used for attachment. And this is going to be a specialized pilus. It also is used for attachment. In this case, it's used to attach one bacteria to another bacteria. So I'm going to go ahead and label that. That is a sex pilus. And it's essentially going to be a hollow tube. And let's go ahead and put our F factor in here. And again, this F factor is double-stranded DNA. I drew a single line, but it's double-stranded DNA. And what's going to happen here is one of these strands is going to become unbound. It's going to become disconnected. And we're going to transfer that strand through the sex pilus to the other bacteria. Now let's go ahead and draw a third diagram and show the finished product. Again, here's our chromosomes. Here's the F factor. And one of the strands, remember this is double-stranded DNA, one of the strands is going to be threaded through the sex pilus into the other cell. So there it is right there. And once that strand gets into the other cell, both of these F factors are now going to reproduce. They're going to remake that second complementary strand to become double-stranded again. They're going to replicate a complementary strand so that they're double-stranded. And when it's all said and done, not only is this guy going to remain F positive, but the F negative cell now becomes F positive, and it can transfer that genetic information to another bacteria, and the process continues. And that's going to be it for bacterial conjugation.